Good afternoon, everybody. I am Kathleen van Craenenbroeck, and first of all, I would like to thank the Dean for this opportunity to present our research here. I would like to start with a small history of the lab. So the LEGIS lab, this is the abbreviation for Laboratory of Eukaryotic Gene Expression and Signal Transduction. And this lab was started up by Professor Hageman. So after a while, there were like 3 independent research groups. So there was a group focusing on nuclear receptors that was coordinated by Caroline de Bosser. Then there was a group of Wim van den Berghe who focused on phytochemicals and the role in the treatment or the prevention of several diseases like cancer. And then there was my group that focused on GPCRs. With the retirement of Professor Hageman inside, things became a bit difficult, and Caroline de Bosser left the lab and moved to the Faculty of Medicine together with her group. And then also Wim van den Berghe, he got a full-time application at the University of Antwerp, so he also left the lab, but he is still associated with our group, and his topics are now coordinated by Karen Heining. But the main focus for the moment is on G protein coupled receptors. So we changed the logo a bit to this, and we also changed the name a bit because it's not like all eukaryotic genes anymore. We will focus on GPCRs. So GPCRs are 7 transmembrane passing receptors that couple to G proteins, and they form the largest group of membrane receptors, and they are like almost everywhere in your body. They are responsible for vision, they are responsible for taste, they control your mood, they also control movement, and so there are really a lot of GPCRs, and there are also a lot of ligands that can bind to these G protein coupled receptors. Those ligands can vary from very small. For example, a photon can induce a conformational change in this receptor, but also rather big molecules, such as endorphins, can bind to these molecules. Endorphins are those molecules, it's like your body-owned morphine. So if you participate in the 10 miles of Antwerp, it's possible that after 2 kilometers you are really in pain and you think you will never survive, but then your body starts to produce endorphins. They bind to opioid receptors, and you will feel good and you will finish within a nice time. If you don't make those endorphins, you will suffer and you are less lucky, of course. Yeah. Other well-known ligands for these GPCRs are, for example, dopamine, serotonin, acetylcholine, glutamate, or GABA. Yeah. And they regulate a lot of important processes, so I challenge you to have a look at your medical cabinet this evening, and I'm pretty sure you will find some drugs that will bind, that will activate, or that will block one of these GPCRs. So for example, nowadays a lot of small children get those inhalers. So there the active compound is salbutamol. That's an agonist of a beta-adrenergic receptor, and it will help with breathing. Probably you can also find motilium. There the active compound is domperidone. That's a blocker, an antagonist of dopamine receptors. When you get older, it's possible you have some heart problems, and then you take what we call beta blockers. Beta blockers are antagonists of beta-adrenergic receptors. And then when you get even older, it's possible that you get some neurodegenerative diseases, for example, Parkinson's disease, and then you will have to take an agonist to stimulate your dopaminergic transmission. So this is just a very small list, but up to 50% of all the drugs that are sold in the pharmacy nowadays will activate or inhibit this kind of receptors. And here is just a list with some hallucinants, with some fascinating sales numbers. So you see that there is a lot of interest in this kind of receptors. And although there have been major breakthroughs in this research, I have the feeling that the full therapeutic potential of these GPCRs are not reached yet, just because we do not know exactly what is happening on a molecular level here with these receptors. So there is still a possibility to learn more about these GPCRs. So what is known for the moment concerning the molecular signaling? So a ligand will bind, it will activate the receptor, it causes a conformational change, and this will lead to activation of a G protein. So G protein consists of 3 subunits, alpha GDP and then beta and gamma. Upon activation, the GDP will be exchanged for GTP, and the alpha GTP will dissociate from the beta gamma dimer. So both the alpha GTP and the beta gamma dimer can have an effect on other proteins. For example, they can open ion channels, or they can lead to different differences in the concentration of second messengers. And that can then cause a signal transduction pathway going to the nucleus, having an effect on gene expression, and then you have a long-term regulation. I told you that those GPCRs are like everywhere in your body, so we had to narrow down a bit our research. So we are focusing on GPCRs that are important in neuronal signaling. So here you have an oversimplification of a neuron. So you have the dendrite, the cell body, and then your axon. And if you think about neuronal signaling, you can divide it in 2 parts. So you have an electrical part and a chemical part. And the movement of a signal from the dendrites through the axon will be an electrical signal. So there is a change in charges, a change in ions that is going through the membrane, and that moves very fast to the end of the neuron. When that electric signal comes at the end of the neuron, it will be translated in a chemical signal, because an electrical signal cannot just jump from one neuron to another neuron. So here it's a bit described in more detail. So the electrical signal comes, there is an opening of ion channels, and here also ion channels for calcium will open, calcium comes in, and then these vesicles that are full with ligands, with neurotransmitters, will fuse with the presynaptic membrane. The ligands come into the synaptic left, and they can bind postsynaptically. If the binding is so strong, it's possible that a new signal is generated postsynaptically, and that's then again an electrical signal. The electrical signal goes very fast, and it's hard to regulate that. But here, that chemical signaling part goes slower, and it's also possible to regulate this into more detail. So, for example, if you have a dopaminergic neuron, that means there is dopamine in those vesicles. So, upon receiving an electric stimulus, dopamine will be released in the synaptic left and can then bind to dopaminergic receptors. So here you see some drugs that can interfere. For example, in Parkinson's disease, you will have a higher concentration of your dopamine, so you will add L-dopa. You cannot give dopamine to Parkinsonian patients because dopamine cannot cross the blood-brain barrier. You can also, for example, in schizophrenia, the dopaminergic signaling is too strong, so then you have to block the dopamine receptors. Another well-known drug is cocaine. Cocaine binds to the dopamine transporter and inhibits the transport, so you will have more dopamine in the synaptic left, so more signal. So you will feel a bit high. So you understand that with this chemical signaling, there is much more regulation possible. And we focus on serotonin and dopamine receptor. Serotonin receptors are important in cognition and mood. Think about Prozac. Prozac blocks the uptake of serotonin, so there is more serotonin in your synaptic left, so more signaling. And serotonin receptors are also important in migraine. Then the dopamine receptors are important in motor control. Think about the Parkinson. And cognition, learning and reward, and in addiction, and then also mood. About the reward system, like if you eat, you have to eat because that's good for your survival, so then dopamine will be released, so you get a reward because you eat. But those natural rewarding systems like eating can also be replaced like with nicotine or alcohol, and then you are more in the field of addiction. And then we also study some other GPCRs, but always in the frame of interaction between with the dopamine receptors. And opioid receptors are important in pain and addiction, and then acetylcholine receptors in cognition, mostly Alzheimer's disease, and metabotropic glutamate receptors are also important in neurodegeneration. Okay, so we are the lab of GPCR expression and signal transduction. So we have several topics going on in this field. So I will start with the story where we studied GPCR expression. So membrane proteins in general are folded in the endoplasmic reticulum. So here you see the endoplasmic reticulum, and there is quite some control. So only proteins that are folded correctly will be able to pass to leave the endoplasmic reticulum and continue their journey via the Golgi to the plasma membrane. If a protein is not folded correctly, it will be retained in the endoplasmic reticulum, and it can be or degraded in the proteasome, or it can go back, unfold and fold again. So we found that if you add ligands that can bind to, in our case, dopamine receptors, that you can enhance this folding. So for example, here you see the expression of dopamine receptors. If you add dopamine, we cannot see an effect. That's because dopamine cannot cross the blood-brain barrier, it's not membrane permeable. So here you do not see any effect. If you add quinperol, that's an agonist, or if you add antagonists that are membrane permeable, they can go to the ER, help with the folding, and increase the expression. So that's why we call these ligands pharmacological chaperones, because they help in the folding of the protein. So we measured this with ligand binding assays or via Western blot immunodetection. We also had a folding mutant, so somebody asked to make this mutant. We could not express it because there was like a problem with the folding. If we add then drugs, pharmacological chaperones, we clearly see a good expression of this receptor. So this was a nice story, but it was all in an artificial system. So we wanted to know if this was also occurring in vivo. As we do not have the tools for this, I contacted Professor De Vos from the Faculty of Pharmacy, and Nick van Laken is a PhD student who is testing this in vivo. So Nick labeled raclopride, a ligand that detects dopamine receptors, with C11, and then they do a PET scan of the animal. Yeah. The biggest advantage is that you can work in a non-invasive way, so you do not have to kill the animal. You make an image, you wait, you treat the animal, and from the same animal you make a new image. Yeah? So So here we tested that pharmacological chaperone effect, but unfortunately from the first experiments we could not see that the drug could enhance the expression of the receptor. It's possible that there is no pharmacological chaperone effect, but it's also possible, because you work in vivo, that other effects are masking this chaperone effect. For example, if you have the release of dopamine, there can be competition with raclopride, and then you will have less bending. But Nick still has some time to solve or to answer the questions, so this is still going on. To get some new ideas about GPCR expression and signal transduction, we thought it would be good to look for new interacting proteins, just to get new insight, new information. And I will tell you two stories about these proteins. So one story is about the dopamine D4 receptor. And this is a particular receptor because in its third intracellular loop, there is a repeat region, and this region can repeat it from 2 until 11 times. And the most common variants are D42, D44, and D47. And this one, persons with this repeat allele, they show a predisposition to develop ADHD. Yeah? So we found in our screening a protein that binds specifically to this repeat region, and that protein is causing a post-translational modification of the receptor. So it adds an extra group on the receptor, a ubiquitin group, and that can then give rise to differences in signaling, differences in trafficking. So here you see a Western blot, so we express different forms of the D4 receptor, so for 2, for 4, and for 7, and here the same, but in the presence of KLHL12, that interacting protein. So if we then look to that post-translational modification, to the ubiquitination, we see if KLHL12 is present, that there is more ubiquitination, so more black, more ubiquitination. Yeah? Also for the D44, we see an increase in ubiquitination. For the D47, so the one associated with the development of ADHD, we do not see this ubiquitination. So this was really something very interesting, because until now there were no differences published between those variants. So this is then the model. So you have the repeat region, the interacting protein, and then here other proteins that are involved in the ubiquitination of the receptor. Most of the time, this post-translational modification gives rise to degradation, but this we could not show. So Camilla is involved in this research, and she is now investigating the role in signaling. Another interesting finding was that normally ubiquitination is added on a lysine residue, but here we also found that serine threonine and cysteine residues could be ubiquitinated. So this work was initiated by Peter and Bea, and now Camilla is continuing this. Our research also led to the insight that GPCRs can interact with each other. So this is the initial paradigm where you have a ligand, the ligand binds to your GPCR, and you get the activation of a G protein. But this protein is there together with other GPCRs, so it can form like homodimers, two red ones, or heterodimers, for example, a red and a green one, or oligomers. So when this ligand will bind to this receptor that is in complex with other GPCRs, it's possible that the binding of the ligand will change, but it's also possible that the signal transduction will differ. So this makes the whole view on GPCRs much more complex, and it gives also rise to new complexes that are not investigated yet, and that can be new therapeutically active, of interest. So we have already published quite some studies about this, and we will also continue with this in the future. I will show one example, just also to show you the techniques we have in the lab, and then some pictures with some data. So what do we want to know and how do we study this? So first of all, we have our in vivo question, and here the in vivo question came from our collaborators in Malaga, where they saw that in the striatum, if they treat the rat with an agonist of the dopamine receptor, that the expression of the mu-opioid receptor went down. They also saw on the expression of this protein, so there is an increase when you add morphine, but this effect is completely abolished when you stimulate dopamine receptors. So from this we know that both receptors are expressed in the same cell, and that there is an influence on each other. So as we are studying protein-protein interactions, GPCR dimerization, we thought it would be useful to study if there is an interaction, an immediate binding of mu-opioid receptors with dopamine receptors. So to obtain evidence for that, we perform co-immunoprecipitation assays, breath assay, and proximity ligation assay. So the idea of a co-immunoprecipitation is that you have a cell expressing, for example, a blue, a dopamine receptor, and then here a red, let's say an opioid receptor. You lyse the cells, you add an antibody that recognizes, for example, here the dopamine receptor. Then you add a bead. The bead will bind to the antibody binds to your dopamine receptor, and if you will precipitate this, it will not only be the dopamine receptor that will be precipitated, but also all the proteins that bind to this, this protein of interest. So then you have precipitate, you have isolated a whole complex, and then you can analyze the proteins in that complex. So that is then analyzed in a Western immunodetection plot. So here you see the expression of the dopamine receptor, the expression of the opioid receptor. We isolated the dopamine receptors, and then we look if the opioid receptor is present. So this was the case. So this was a good start to continue and to get extra confirmation. One very interesting method to study protein-protein interactions is via BRET. So BRET is the abbreviation of bioluminescence resonance energy transfer. So you get a transfer of energy from one molecule to another molecule. So here, for example, you fuse your dopamine receptor to luciferase. And then you fuse your opioid receptor to yellow FP. If you give a substrate, the luciferase will convert the substrate and will emit light. And that light can then excite yellow FP, but only if those proteins are close to each other. If the distance between these two proteins is bigger than 100 angstrom, it will not be possible to excite this molecule just with the light that is coming from your luciferase. So if there is an interaction, your energy transfer can occur. And this is one of the results. So you keep the concentration of your donor constant, and then you add increasing amounts of your acceptor. And if you get such a hyperbolic curve, that means that these proteins are interacting. If you get such a line, there is no interaction. Yeah? It's a very artificial system. I agree on that. And if we want to prove things in vivo, this is not that useful. But it's interesting because you can determine the affinity of the dimerization between these two, and you can compare them. You can also develop tools like bivalent ligands or interfering peptides and test them with this method. We also wanted to get some in vivo or evidence, so we did co-immunoprecipitation in neuronal cells. That gave some nice results, and then we also performed a proximity ligation assay. This assay is based on different antibodies. So you have an antibody recognizing your dopamine receptor raised in mouse, and then another antibody recognizing your opioid receptor and raised in rabbit. And if these two are close to each other, the secondary antibodies can form such a rolling circle, and then this amplification can be visualized. This technique is not running in the lab for the moment yet, but we hired a new student, and she will go to the Karolinska Institute to learn this technique. So here you see a slice of a rat brain, and those red dots you see here, that means that there is amplification, so there both proteins are close to each other. Then in other regions that are our negative controls, you do not see those red dots, so there was no amplification possible, so no interaction. So that were our interaction studies, and this is a good start, of course, but of paramount importance is the functional role of this interaction, because okay, they can be together, but if there is no effect on ligand binding or there is no effect on signal transduction, who cares? So we did different tests. We did different tests on ligand binding, so for example, internalization or different parts of the signal transduction pathway to study the interaction between both proteins. And here you see the results of a phosphorylation assay of P44, P42. So what we do, we treat the cells with an agonist, so we stimulate the opioid receptor, and we get a significant increase of the phosphorylation of our protein. But if we stimulate the other partner, we do a pre-treatment where we stimulate the dopamine receptor for 30 minutes, and then we do a treatment of 5 minutes. This is significantly decreased, and this effect is called desensitization. We also see this desensitization effect on receptor internalization. So here you have the opioid receptor expressed on the membrane. So if you stimulate these cells, you will see dots in the cell. So that means that the receptor is not on the plasma membrane anymore, but that it's internalized. Yeah? And if you then do a co-stimulation with an agonist of the dopamine receptor, this internalization is less. So this is visualized then here. So this also indicates that there is a crosstalk between both receptors. So this was also done in artificial system. So we wanted to make the shift to native tissue. Do we also see that cross-desensitization in native tissue? And this was in collaboration with Professor Lefebvre from the medical department, because they do assays in hippocampal slices and they measure the release of acetylcholine. So you have a hippocampal slice, you stimulate, and then you measure the release of acetylcholine. And then after a while you stimulate again, and normally in your control your S1 and your S2 should be the same. So the release of acetylcholine here and here should be the same. If you then stimulate in between with, for example, an agonist of the opioid receptors, you can measure the effect on acetylcholine. So here you see that, for example, when you stimulate the cells with an agonist of receptors, that there is a decrease of acetylcholine. If you treat the cells, the hippocampal slices, the whole time with an agonist for the opioid receptor, and then you do this short treatment, then you lose this significant effect. So this can also gives us an idea that there is some desensitization going on in the hippocampus. So this is a scheme how we think that those GPCRs are interacting with each other. So if there is only one GPCR, you add your agonist and you get a signal. But if there is another agonist present, it's possible that this agonist works as a negative allosteric modulator. So just the presence of that interacting partner can already give rise to a smaller signal, although you use the same agonist, the same concentration, the same induction period. If you then add an agonist of this receptor, the effect, the negative allosteric effect on your first GPCR will even be bigger, and your signal will become even smaller. Yeah? So that is what we mean with the cross-desensitization. So for this part, we can conclude that there is that we showed interaction in vitro and endogenous, and then in function, we saw cross-desensitization in vitro and then also ex vivo. And from the collaborators in Spain, they also see this in vivo. So they are studying pain and then also pain in relation to addiction. Yeah? So that means that you can stimulate your opioid receptors and have an enhancement in the pain control without the addictive effect. Yeah? So, and this work was mainly done by Jolien, and then these are the collaborators in the Karolinska Institute. And then the last slide I would like to spend on our future perspectives. So, because there are still some gaps in our knowledge, and for the moment a lot of studies about GPCR dimerization describe similar experiments and so, but we need more data for the in vivo evidence of the dimerization. So we want to develop new tools to study this dimerization. So one of the tools are interfering peptides. So they should interfere with the dimerization so that you can study what the effect is of the dimer and then the two proteins just apart, separately. Then we also started with the development of bivalent ligands. This is in collaboration with Serge Van Craenenbroeck from the pharmaceutical faculty, and Ming Cheng is the PhD student there. He already made some of those bivalent ligands, and he is now in the lab to test them for ligand binding and for effect on signaling. So with this idea, that cross-desensitization effect, and so we also thought maybe the pharmaceutical industry can be interested in this, because like here is one of the ideas, like for Parkinson, people get L-DOPA, so you get the stimulation of the dopamine D2 receptor, but this receptor is not alone. There you have, for example, an interaction with the glutamate receptor, and this blocks some effect of the dopamine receptor. So it works as a negative allosteric modulator. So if you then can have a multi-therapy where you have an antagonist of the mGluR5 together with lower doses of the L-DOPA, you can get the same effect. Also, if it would be possible to develop bivalent ligands in which you block this, you activate this, you can go to a more specific therapy. So with this idea, I went to Tech Transfer, and they arranged a meeting with Janssen Research Foundation where they were really enthusiastic about the idea. but they had a lot of questions, and we also had a lot of questions, so we assembled all our questions and 4 work packages. And in one work package, we want to validate those receptor dimers, and in other, we want to know more about the protein-protein interaction. Also, if we can do a prediction, can we predict if 2 GPCRs can interact with each other? What is the interaction domain? Also, what is the stoichiometry? Is it 1-1 or like 1-2 or 1-3? And then we also have a whole work package on the functional studies, so the ligand binding and signaling and the neurotransmitter release. And it's a combination of biochemistry, biophysics, but also molecular biology and then physiology. And for the physiology part, we want to study behavior in certain diseases, and then we want to test multitherapy, interfering peptides, and bivalent ligands. So we assembled a consortium, so we will do quite some parts of this, then together with Savas, who will be responsible for the protein-protein interaction part. And then this is Francisco Ciruela. He is a guest professor in our group. And then the behavioral studies will be performed in the laboratory of Peter de Deen from the University of Antwerp. He's an expert in Alzheimer's disease. And then we will work together with Paul Boon here from the UZ in Ghent, who is an expert in epilepsy. So at the very far future, the idea is if we have a disease in which GPCRs are important, that we screen those GPCRs who can interact with each other. Are they important in the different stages of the disease? For example, in Alzheimer's, you have models, so you can take from several stages brains and then study the protein-protein interaction. So this is then the screening platform, and at the end we want to come to like 1 or 2 GPCR interactions and then develop bivalent ligands or interfering peptides. But that's like for the very far future. And then a second screening platform we are developing is a functional screening platform where we have the ligand binding assays and then the signaling assays. And here we want them to test the role of the interfering peptide, the role of multitherapy, the role of bivalent ligands. And then I would like to thank all the people from the lab and the people with whom we collaborate, and then also the institutions that support us. And I also would like to thank you for your attention. And if you see opportunities or things that can be helpful, just contact us or visit our website. Thank you.
Very much. We have time for a few questions.
Yeah.
Yes, please.
Do you have any experimental evidence that if your receptor interact, that one receptor is signaling towards the other and the other one actively signaling downstream?
The only, here the experiment I show here with the, here, normally in this assay, your peak is after 5 minutes. And after 15 minutes, you do not have any effect anymore. So if we treat the cells for 30 minutes, then normally you do not see any effect on phosphorylation of MAP kinases anymore. So if you then treat with a DAMPO, so you stimulate your opioid receptor, that goes via the opioid receptor, and then the effect normally should come via the dopamine receptor. Is that somehow? Okay.
any studies being carried out in terms of trying to cross-link these, chemically cross-link them together and isolate them as a complex?
No, not like for the moment, but we have plans with Savas to do indeed some cross-linking studies. Yeah.
Because that would be a way to isolate the complex,
Yes,
make
yes.
them stable and then...
Yes, that was also a remark we got on the project proposal that, yeah, if there were like more experiments to be 100% sure, because with the assays I show, there is like always a chance that there is another protein like in between. Yeah. So.
And in that sense, is it possible to express these GPCRs with a slight modification, or do you need to rely on exogenously and cross-linkers?
Maybe it's possible to make like a fusion protein with it. That's maybe possible, but this I have not seen before, but it's maybe something to try. Thanks for that.
Yes?
Perhaps a more practical question. I've noticed for these studies here that you used stable Hector 9.3 cell growth. Now, is there a reason why transient expression doesn't work in these setups?
Because like phosphorylation of MAP kinases is already like an indicator for stress. So if you work transiently, you bring in the DNA and then 2 days or 3 days later you have to do your assay, and then the cells are still rather stressed. So in our hands it didn't work. So we have stable ones, and there we see a nice curve. So that's just because it didn't work in the other setup. Yeah.
Do you know why the second repeat domain is not or much less repeated?
No, we do not know yet. Camilla also investigated if the whole complex was still there with the E3 ligase and so everything should be there. But why it's not ubiquitinated, we do not know.
Any more questions? If not, well, thank you again, and let's thank those people.